animal-based personalized nutrition approach. So to begin with, what is Nutrigenetics and Nutrigenomics? So Nutrigenetics deals with the study of the impact of the DNA sequence variation on chronic disease outcomes in response to a particular diet. That is, in response to a particular diet, such as a high-carb diet or a low-protein diet, how are my genes going to respond to the development of a particular disease? So I'm basically trying to understand what is the relationship between the genetic variations and the dietary factors in relation to the development of a particular disease. And that field is what you call as Nutrigenetics. And Nutrigenomics, as you can see from the name, omics means expression, genomics means gene expression, and Nutrigenomics deals with the impact of the nutrient on gene expression. So this is what we study as part of the Nutrigenomics. You're looking at the gene expression level. If I consume a high-carb diet or a high-protein diet, what are the genes that are expressed in response to a particular diet? So that study is Nutrigenomics. So the field of Nutrigenetics and Nutrigenomics are completely two independent fields of research. Of course, the findings from Nutrigenetics and Nutrigenomics are essential to implement personalized nutrition, where an optimum diet could be tailored according to that individual's genetic makeup. That is, personalizing an individual's diet according to that individual's genetic makeup requires the findings from Nutrigenetics and Nutrigenomics. Now let's have a look at what is Nutrigenomics. As I said, it is a study of the role of nutrients in gene expression. So it all starts with the food that you're consuming. So the food that you're consuming has got several nutrients. It can have macronutrients, micronutrients. So all these nutrients need to be utilized by your body. And how is this utilization taking place? It needs to be taken up by the cells, which are present in different parts of the body. And the nutrient enters the cell. And once it enters the cell and reaches the cytoplasm, there are several proteins which are waiting for the nutrient. And these proteins are nothing but the transcription factors. So there are specific transcription factors for specific nutrients. For example, if you look at these transcription factors, PPAR is a transcription factor for a nutrient such as fatty acid. And CHREBP is basically a transcription factor for the nutrient which is rich, if you're consuming a diet that is rich in carbohydrates. And VDR stands for vitamin D receptor, so which is a transcription factor which is specific for vitamin D. So the main function of the transcription factors is basically to bind to the nutrient. So there are specific transcription factors for specific nutrients. For example, if you look at these transcription factors, PPAR is a transcription factor for a nutrient such as fatty acid. And CHREBP is a microphone factor for the nutrient. Yeah, thank you very much. Yeah, so the transcription, the main function of the transcription factor is basically to go and bind to the nutrient and take the nutrient to the nucleus where we have the genetic material, which is the DNA, deoxyribonucleic acid. And the DNA, the whole human DNA has got about 20,000 to 25,000 genes. Within these 25,000 genes, you have different genetic variations. So these transcription factors, they bind to the nutrient and take this nutrient to the nucleus and goes and bind to a very specific and a very unique region in the gene. And that unique region is nothing but the promoter region. So this nutrient with the transcription factor binds to the promoter region and initiates the process of gene expression or the gene regulation. The first step in the gene regulation is the transcription, where the genes are converted to the RNAs or messenger RNA, ribonucleic acid. And these RNAs are produced in the nucleus. And once they are produced, they are sent outside of the nucleus to the cytoplasm, where they undergo the protein synthesis process or the translation, where the RNAs are converted to proteins, and proteins, either they are packaged into molecules to get released into outside of the cell to perform multiple functions, or the proteins are utilized by the cell for various other activities. or the proteins can also be metabolized to produce several metabolites. So in response to a particular nutrient, there are certain RNAs that are synthesized, there are certain proteins that are synthesized, and there are certain metabolites that are produced. So the study of the production of the RNA transcript in response to a particular nutrient is what you call as a transcriptomics. That is, you're looking at the expression of the RNA transcript in response to a particular nutrient. And the study of the expression of the proteins in response to a nutrient is a proteomics. And the study of the expression of the metabolites in response to a nutrient, and that is what you call as a metabolomics. So all these fields of omics approaches, genomics, transcriptomics, proteomics, metabolomics, they all come under the field of Nutrigenomics. So why do we have to study all these omics approach? That is a very big question. So for example, if you're consuming a high-fat diet, which is a unhealthy diet, what happens? There are certain genes which are not expressed, and there are certain genes which are overexpressed. But if you consume a healthy diet, there is a normal expression of all the genes. So when you consume an unhealthy diet, there is an abnormality in the gene expression levels. And that change, that variation in the gene expression level, and that variation becomes a molecular biomarker. So once you know that that is a molecular biomarker, that a person is consuming an unhealthy diet, so by modifying that unhealthy diet, that is changing the dietary pattern, can correct the gene expression levels. So that becomes a strategy in preventing the development of the disease. For example, if you have been consuming an unhealthy diet for a very long time and your body is exposed to all the abnormalities in the gene expression for a very long time, that long-term exposure can eventually increase the risk of you becoming obese or becoming diabetic or even developing cardiovascular disease. So you're basically trying to correct the gene expression levels based on changing your dietary pattern. And you're basically focusing on preventing the development of the disease. And this is what you study as part of the Nutrigenomics study. So this is looking at the gene expression. Now looking at the Nutrigenetics, which is nothing but the gene-diet interactions. That is how your genetic factors are interacting with the environmental or the lifestyle factors such as diet or physical activity and contributing to the development of a particular disease, such as obesity or diabetes or cardiovascular disease. So looking at the gene-environment interactions on obesity, so there are two important factors. One is the lifestyle factors and the genetic factors. So the development of obesity depends on the interaction between the genetic susceptibility and the unhealthy diet and the lack of physical activity, which both of which equally contribute to the development of obesity. So obesity is basically a complex disease, a multifactorial disease where there are multiple genetic factors and multiple lifestyle factors such as dietary factors and physical activity levels and various other unknown environmental factors which all interact together and contribute to the development of obesity. But the most important thing is only if you provide an obesogenic environment, you develop obesity. That is, even if you carry a very high genetic risk, you can still overcome that genetic risk by modifying your environment. And that's why we say genes load the gun, but the environment pulls the trigger. So you might carry the genetic susceptibility, but unless you provide that obesogenic environment, you're not going to become obese. So this is the underlying basis on which we do all these nutrigenetics studies. So what is a genetic variation? So let's take an example of this individual has got this DNA sequence. We know that every individual has got 2 copies of each gene. So the 2 strands that you see basically, they are basically the 2 copies of a particular DNA sequence. And this population has got, for example, 5 individuals, for example. And you can see that 99.9% of the DNA is identical and 0.1% carries all the genetic variation. So what is that genetic variation? So if you look at this particular sequence, you can see that some individuals carry T and some individuals carry A. So there's a change from one nucleotide to another nucleotide. This change from one nucleotide to another nucleotide is what you call as a single nucleotide polymorphism, or in short, SNP. So depending upon whether you carry T or A. So individuals who carry A are the individuals who carry the genetic variation. So depending upon whether you carry 1A or 2As, you can classify individuals into 3 different groups. So 0, 1, and 2. So individuals who do not carry any copies of the genetic variation, that is, those who do not carry any copies of the A nucleotide or the genetic variation, they are classified as 0 copy individuals. That is, they don't carry any copies of the genetic variation. So if you carry 1 copy of the A, that is 1 copy of the genetic variation, those individuals are classified as 1 copy individuals. And if you carry 2 copies of the genetic variation, that is 2 copies of the A, they are classified as 2 copy individuals. So for a given genetic variation, you can classify individuals into 0, 1, and 2 copies. In general, individuals who carry 2 copies, they have increased genetic risk of developing a disease compared to 0 copy and 1 copy individuals. So what is that genetic risk? So if you look at individuals carrying 0 copy, it means low genetic risk. So that means you have decreased risk of developing a particular disease. So 1 copy individuals, that is, you have a moderate genetic risk, which means that you have a moderate risk of developing a particular disease. But if you have 2 copies, it means high genetic risk. It means you have very high or increased risk of developing the disease. But this is where you need to focus. Just because you carry 2 copies, it doesn't mean that you have increased risk. That is, it doesn't mean that you're destined to become diseased. You can still overcome this genetic risk by modifying your lifestyle. That is, consuming healthy foods and modifying your physical activity levels, that is increasing or improving your levels of exercise. So that can overcome the genetic risk of becoming obese or becoming diabetic or developing cardiovascular disease. So this is the underlying basis on which we do the Nutrigenetics and Nutrigenomics studies. That is, even if you carry 2 copies, you can still overcome the genetic risk by modifying your lifestyle. So now looking at some of the nutrigenetic studies that have been carried out in developed countries. So it all started off with the discovery of the obesity gene. So this gene was discovered exactly 13 years ago by 2 independent genome-wide association studies, which were carried out at the same time in 2 different populations, one in the UK population, one in Spanish population. And both the studies clearly show that individuals carrying 2 copies of this obesity gene, they have increased risk of becoming obese. So once this data was published, people started to have that fear. So people started to believe that if they carry 2 copies, they are destined to become obese. So to clear this fear from the minds of public, genetic epidemiologists decided to test whether consuming a healthy diet and doing more levels of physical activity, can that overcome the genetic risk of obesity? So that was a time when Nutrigenetics and Nutrigenomic studies started to come into picture. So what is so special about this obesity gene? This obesity gene is otherwise called as the FTO gene. FTO stands for fat mass and obesity associated gene. The genetic variations within this gene have been shown to be strongly and consistently associated with obesity traits in various populations, in different ethnic groups, in men, women, adults, and also in children. So having seen such a strong effect of this gene on obesity, the first question that comes to our mind is, can this effect be modified by a lifestyle factor such as diet? So briefly looking at the hypothesis behind this gene-lifestyle interaction, so we know that there are several gene variants for different obesity traits. So the question is whether this genetic susceptibility can be modified by a dietary factor. So I'm going to give you a couple of examples from my nutrigenetic studies that I've carried out in the UK, US, and the European populations. So the first example is a study that was done in 11,000 individuals from 5 different European countries. So where we collected the samples from 5 different countries and we carried out a meta-analysis, and all these individuals were followed up for 3.5 years. So which was basically a longitudinal study design. So here we looked at several genes and several dietary factors, but we found a very interesting interaction between this obesity gene variation and glycemic index on weight gain. So in this graph, you can see that on the x-axis, you find the glycemic index. On the y-axis, you find the weight change per year. And the three lines are basically indicating the individuals carrying 2 copies, 1 copy, and 0 copy. And as you can see that as glycemic index increases, the annual weight change also significantly increases for those who carry 2 copies. Whereas the annual weight change did not change significantly for individuals carrying 0 copy and 1 copy. So which clearly shows that if you carry 2 copies and if you're consuming a diet that is high in glycemic index, then you're going to gain more weight compared to people who carry 0 copy and 1 copy. So in terms of developing a personalized diet for individuals carrying 2 copies, the personalized diet would be a diet that is low in glycemic index. That is, lowering the dietary glycemic index will be the best way to prevent weight gain in those individuals who carry 2 copies of this obesity gene variation. So the next example that I'm going to give you is a study that was done in the US population in nearly 37,000 individuals where fried food consumption was focused. So the individuals were stratified into 3 different groups based on the frequency of fried food consumption. Those who consume fried foods less than 1 time per week, 1 to 3 times per week, and more than 4 times per week. That is, those who were consuming fried foods quite frequently, almost every day. So in the first study, which was done in nearly 9,000 individuals, you can see that individuals consuming higher amounts of fried foods within a week, that is a white bar, those individuals had very high genetic risk. They had increased genetic risk compared to individuals who were consuming more of the healthy foods. And the second study also showed a similar finding, and also the third study. So we pooled all the samples together. We carried out a meta-analysis, and the pooled meta-analysis clearly showed that individuals consuming fried foods more than 4 times per week, or those who are consuming unhealthy foods quite frequently, they had increased genetic risk. And the third example is about a study that was focusing on Mediterranean diet. So Mediterranean diet is considered to be a healthy diet, which is rich in omega-3 fatty acids, fruits, vegetable, and olive oil. So they are all considered to be healthy foods. And we looked at whether this Mediterranean diet, consuming Mediterranean foods, can that overcome the genetic risk of obesity? So this study again was done in 11,000 individuals in a longitudinal study design. And we identified that individuals who carried 2 copies of the obesity gene variation, they gained more weight compared to individuals who carry 0 copy or 1 copy. And next, we introduced the Mediterranean diet into our analysis, and we found that individuals who are consuming more amounts of Mediterranean foods over a period of time, they alone were able to overcome the genetic risk of gaining more weight. And this study again clearly pointed out the fact that consuming a healthy diet can overcome the genetic risk of obesity. So all these studies clearly shows that the genetic factor and the lifestyle factor, they do not work independent of each other. Rather, they work together, interact together, and contribute to the development of obesity. So the take-home message is basically, even if you carry 2 copies, you can still overcome the genetic risk of obesity by adopting a healthy lifestyle. Now, let me move on to the Nutrigenetic studies in lower middle income countries, that is basically looking at developing countries. So that was one of the main focus point of my research projects. So I started off with this genuine collaboration 6 years ago. Genuine stands for the gene-nutrient interactions. And this collaborative effort has been started with the objective of implementing Nutrigenetic studies for the first time in several lower middle income countries. And these are the countries in which this genuine collaboration has been implemented. That is, the Nutrigenetic studies have been implemented. And these are some of the publications from genuine collaboration. And these are the funding organizations that have supported me in implementing the Nutrigenetic studies. So why Nutrigenetic study is so important in lower middle income countries? So the first important thing, or first important reason, is the different genetic makeup. My DNA is different from your DNA, and your DNA is different from your family's DNA and your friend's DNA. And that is the reason why we all looking different from each other. So the DNA is a very unique component, not only at the individual level, even at the population level and ethnicity level. You can easily identify that whether a person is from South Asia or from China or from the UK or from Europe or from Africa. So you can easily identify a person and you can differentiate different ethnic groups. And the main reason for that is your genetic makeup. So the difference in the genetic makeup is the reason why we all have different phenotype. Phenotype is a physical representation of an individual, and the genotype is a genetic representation of an individual. And the differences in the genotype contributes to the differences in the phenotype. And this is one of the reasons why I wanted to focus on each and every population, because findings of Nutrigenomics study from one population is not applicable to the other population. You cannot use the findings from European population to implement personalized nutrition in India. So you have to do the Nutrigenomics study in the Indian population to implement personalized nutrition in Indian population. So that's the reason why I wanted to implement Nutrigenomics studies in each and every lower middle income countries. And secondly, unique and varying dietary patterns. If you take the Indian population, the Indians have a very unique diet, which is quite different from and quite distinct from other countries. Whereas if you look at the North Indian and South Indian population, you can see a very significant varying dietary patterns across North and South India. So this is the reason why you need to do a Nutrigenomics study, not just in South Indian population, but also in the North Indian population. So this makes it very unique for each and every ethnic group and also for each and every population. And thirdly, increased prevalence of diabetes and obesity, not just India, all over the world. Globally, we are facing the double burden of obesity. So the prevalence of obesity, diabetes, and cardiovascular disease, it is all increasing in all the countries. So without Nutrigenetics, there's no future for personalized nutrition. And these are the main reasons why I wanted to implement Nutrigenomics studies in lower middle income countries. So let me give you some examples of my studies that I carried out in the Asian parts of the world. So the first study was done in the Indian population, which is the first Nutrigenomics study in India. This study was done in 1,000 people with type 2 diabetes and 1,000 individuals who were controls. And this study was done in the population of Chennai, and where we looked at the interaction between the genetic factors and the dietary factors on various cardiometabolic diseases, but we found an interaction between the FTO gene and the fiber intake on waist circumference. The waist circumference is an indicator of central obesity. So in this graph, you can see on the x-axis, you find the tertiles of fiber-rich foods, that is low, medium, and high intake of fiber. And on the y-axis, you find the waist circumference. And these three bars, the blue stripe and red bars, they indicate 0 copy, 1 copy, and 2 copies. The overall interaction was highly significant. But if you focus on this particular group, that is individuals consuming high amounts of fiber-rich foods, they alone were able to reduce their waist circumference by 1.62 centimeter, despite the fact they carry 2 copies. So what it means is that even if you carry 2 copies, that is, if you carry 2 copies, then you should be having a higher risk of obesity. But what we identified was individuals who were carrying 2 copies, when they were consuming high amounts of fiber-rich foods, they were able to reduce the waist circumference by 1.62 centimeter, which clearly indicates the point that even if you carry 2 copies, you can still overcome the genetic risk of central obesity by consuming a healthy diet, such as a high fiber diet. The second study was done in another South Asian population, which is the Sri Lankan population. Here, instead of looking at one gene, we looked at several genes, a panel of genes. The reason why we looked at several genes is that you know that obesity is a multifactorial disease. It is not caused by one gene, it is caused by multiple genes. So we wanted to look at a combined effect of all these genes on different cardiometabolic disease outcomes. And we tested for the interaction with the dietary factors on different cardiometabolic disease outcomes, and we found an interaction with the carbohydrate intake on waist to hip ratio. The waist to hip ratio is another indicator of central obesity. So in this graph, on the x-axis, you find the tertiles of carbohydrate intake, that is low, medium, and high consumption of carbohydrate intake. On the y-axis, you find the waist hip ratio, and the 2 bars, the red and the white, the red one, high genetic risk, and the white bar, low genetic risk. And you can see that individuals consuming high amounts of carbohydrate-rich foods, typically consuming an unhealthy diet, those individuals, if they carry a very high genetic risk, that is a red bar, they will have increased risk of central obesity or very high levels of waist-hip ratio compared to individuals who carry a low genetic risk. So which means that if you carry more number of 2 copies of different genetic variations, and if you're consuming an unhealthy diet, that will increase your genetic risk of central obesity. And the third study was done in the Indonesian population, which is a Southeast Asian population. And here again, we looked at a panel of obesity genes. But interestingly, we found an interaction with the protein intake on waist circumference. Again, waist circumference is an indicator of central obesity. And this study is quite unique because we carried out this study in an Indonesian population, which is called Minangkabau population. Minangkabau is the world's largest matrilineal community. So we focused mainly on the Indonesian women in this population. So in this graph, you can see that on the x-axis, tertiles of protein intake, low, medium, and high consumption of protein intake. On the y-axis, you find the waist circumference, and the 2 bars, the white and the red bar. The white bar are individuals who have low genetic risk, and the red bar are individuals who have very high genetic risk. So if you look at the individuals consuming low amounts of protein-rich foods, those individuals were able to reduce their waist circumference significantly, despite the fact they carry more or a high genetic risk. So this clearly shows that even if you have a very high genetic risk, you can still overcome the genetic risk of central obesity by consuming a healthy diet, such as a low protein diet. So over here, I'm not stressing on the point that consume low amounts of protein. No, that is not the message over here. I'm not asking you to consume or reduce your protein intake. My message over here is, if you look at the high protein intake group, you can see that individuals who are consuming high protein diet, the high protein diet for this Indonesian population or Indonesian cohort was nearly 140 grams per day, which was a mean intake for the high group. But if you look for the low intake group, the mean intake of protein was around 70 to 75 grams of protein. That 70 to 75 grams of protein is the recommended allowance for protein intake for the Indonesian population. So the low protein intake group is actually the normal intake individual. So which means that if you consume the proteins which are required for your body according to the recommended allowance, dietary recommended allowance, then you can easily prevent yourself from the genetic risk. So that is the message from this Indonesian study. And the next study was done in the Western Asian population, which is a Turkish population. Again, here we looked at the interaction between FTO variant and again protein intake on central obesity. So in this graph, you can see on the x-axis, you find the tertiles of protein intake. On the y-axis, you find the central obesity, and the red bars are individuals who carry 2 copies. And you can see that individuals carrying 2 copies of this FTO gene, they had 3.3 times increased risk of central obesity. That is, if they consume high amounts of protein-rich foods, that is consuming more than 140 grams of protein per day, and carrying 2 copies, they will have 3.3 times increased risk of central obesity. So the main message is that protein consumption is definitely good for your body, but very high protein consumption or very low protein consumption, they are not good for you. Anything that exceeds the limit is toxic to our body. So that is exactly what has been shown in these studies from genuine collaboration. Another interesting study which has been published a month ago, which is relating to the metabolic disease and vitamin D status. It is an interaction between the genetic risk and dietary intake on 25-hydroxy vitamin D concentrations. We all know we need very good levels of vitamin D in our body. So this study was done in the Indian population, which clearly shows that even if you don't have a high genetic risk, but still you need to consume a healthy diet to prevent yourself from developing the disease. So in this graph, you can see that on the x-axis, you find the carbohydrate intake in tertiles, low, medium, and high consumption of carbohydrate intake. On the y-axis, you find the 25-hydroxy vitamin D concentration. The black bar, high genetic risk, white bar, low genetic risk. So you can see that individuals who are consuming low amounts of carbohydrate-rich foods and those who carry a low genetic risk, they alone have higher levels of vitamin D. So just because you have a low genetic risk doesn't mean that you're going to have very high vitamin D levels. No. If you want high vitamin D levels, then you should also consume a low carbohydrate diet. Consuming too much of carbohydrate-rich foods can inhibit the production of vitamin D. So this is a study which clearly showed that even if you have a low genetic risk, you will have to still consume a healthy diet to overcome your vitamin D deficiency status. So all these studies from the genuine collaboration clearly provided the first evidence for the gene-diet interactions on central obesity-related traits, which clearly shows an interaction between the FTO gene variant, that is obesity gene variant, and the dietary intake on central obesity. So if you're interested in knowing more about the genuine collaboration, you can go into this publication in Nutrition Bulletin, which was published in 2017, and this was published in 2020, early this year, which is about the genuine collaboration. So briefly touching upon what is personalized nutrition, as I said before, personalized nutrition is basically developing an optimum diet for an individual according to that individual's genetic makeup. That is the tailoring of the dietary advice to suit an individual based on that individual's genetic makeup. So in order to understand in better terms about the personalized nutrition, the University of Reading, where I'm working at the moment, they carried out a study which is called as the Food for Me study, which is basically a study focusing on the consumer attitudes towards personalized nutrition. If somebody is implementing personalized nutrition in a population from a developing country or something, so what is the focus of the people? How do the consumers look at this concept of personalized nutrition? So if you're interested in knowing more about this research on personalized nutrition, you can go into this option research, which will list all the publications in relation to personalized nutrition. So besides Nutrigenetics and Nutrigenomics, there's another important area of research, which is called as a gut microbiome, because the food that you're consuming, it is not only interacting with your DNA, but it is also interacting with your gut microbiome. So the diet-gut microbiome interaction is also another important field of research. So in terms of implementing personalized nutrition, you have to take into consideration of the nutrigenetics, nutrigenomics, and also gut microbiome analysis. So what is the role of nutrigenetics in personalized nutrition? Is it ready for clinical use? So the answer is over here. So it all starts with your diet. So the diet you're consuming, it goes inside your body into three different compartments. The first compartment is what we have already seen today, which is the nutrigenetics. And I've also touched upon the gut microbiome. So where the food enters into your gut microbiome. If you consume healthy foods, your gut microbiome or the gut bacteria will provide all the requirements which are required for normal functioning of your body. But if you're going to consume an unhealthy diet, that's going to have all the outcomes relating to the different disease outcomes. But there is also another important compartment, which is called as the Nutri-metabolomics interaction. That is the metabolites. The diet also interacts with the metabolites, and that area of research is called as the metabolite-diet interaction or Nutri-metabolomics. So all these three compartments, they act in parallel together. That is, they function together. As soon as you consume the diet, these three compartments become really active. So when you're thinking of implementing personalized nutrition, it has to be observed as a three-way approach or a three-way interaction, looking at the metagenome, hyperbolome, and diet interaction. And this interaction is the one that will determine the implementation of personalized nutrition or development of personalized functional foods. So in order to implement personalized nutrition, we need to have the data from all these three compartments. But at the moment, we do not have much research in the field of Nutri-metabolomics. And of course, the field of gut microbiome is progressing and it is very much of an hot field, but we still do not have an evidence of the functioning of all these three compartments together to reach the stage of personalized nutrition. So if you ask me the question, is it ready for clinical application? Technically speaking, it is not. But there are several personalized nutrition centers with the information, whatever is available. They are trying to provide a personalized diet therapy, which has been effective and which has been successful for some people and not for others. So this is where we are in terms of personalized nutrition. So all these emerging human nutrition trends, looking at nutrigenetics, nutrigenomics, proteomics, transcriptomics, gut microbiome, or metabolomics, or systems biology, these are the human nutrition trends which are going to shape the future of food industry in terms of implementing personalized nutrition. So briefly touching upon the concept of the precision nutrition. So in terms how you can translate the finding from nutrigenetics, nutrigenomics to personalized nutrition. So if you want to develop a population-based dietary recommendation or even an individual based dietary recommendation. So it is not sufficient you do just the nutrigenetics, nutrigenomics, metabolomics, and gut microbiome analysis. You need to look at other factors as well, taking into consideration of the family history, race, ethnicity, medication, and various other risk factors, along with the personalized disease stratification markers, that is looking at the lifestyle markers, diet, physical activity, socioeconomic status, eating behaviors, and dietary patterns. And all these things will add up to the level of precision nutrition. So you need to achieve a stage where you can provide the nutrition recommendation based on all these factors together. And that is the one that's going to determine the nutritional phenotype. And that is eventually going to lead to the clinical application of how personalized nutrition could be utilized by using the findings from nutrigenetics and nutrigenomics. So on this note, I would like to mention that the Nutrigenetics is basically a personalized nutrition company in London, where they are focusing on developing genetically personalized nutrition shakes. I'm a scientific advisory board member, and I'm pleased to inform you that we have launched the world's first genetically personalized sports nutrition drink, which has been tailored according to your own DNA. So there are several new nutrigenetics companies and personalized nutrition companies which are coming up. And this one is basically focusing on the personalized sports nutrition drink according to an individual's genetic makeup. So even if you're very lazy, drinking this drink can enhance or boost your genes to become more active. So that is the whole concept about this Nutrigenetics company in London. So the take-home message from my talk is that the genetic factors and the dietary factors, they interact together and contribute to the development of the disease such as obesity. So even if you carry high genetic risk, you can still overcome the genetic risk by consuming a healthy diet and doing more levels of physical activity. So on this note, I would like to mention that the DNA or the diet, so which has got an upper hand in terms of this question, you could say that diet as an upper hand, because the DNA is something that you have right from the day one until the last day of your life. You cannot change your genes. If you're born with 0 copy or 1 copy or 2 copies, you are destined to stay with those genes. You cannot change your genes. You cannot touch your DNA. But what you can change, what you can modify is your diet. You can modify your diet to overcome your genetic risk. So the main role being played in terms of the development of obesity is the diet, the environment. So if you can prevent that obesogenic environment, you can easily overcome the genetic risk of obesity. So I would like to thank all my genuine collaborators and all my funding organizations which have supported me in my work. And finally, thank you all so much for listening to my talk. And if you have any questions, you can send an email to this particular email ID. And I'm available on Facebook and LinkedIn as Vimal Karani. And thank you so much, and happy to take any questions.
Well, that was an extremely informative session with you, Doctor. Shall we move on to the question and answer session? Doctor?
Yeah, sure.
Right, okay. So the first question. What happens to the nutrigenetics in the case of malnutrition children?
Sorry, would you mind typing the questions on the chat box?
Oh, just a second.
Yeah. Or you can read the question again if you need to type all.
Yeah, sure. Okay. What happens to the nutrigenetics in the case of malnutrition children?
So Nutrigenetics is basically, I'm not sure like whether Nutrigenetics, see, Nutrigenetics is more of a study. So the question should be like more about whether you want to know whether the personalized nutrition can be applied to children. So you could do a personalized nutrition therapy for children, but my take on this aspect would be the personalized nutrition is something where you're trying to personalize an individual's diet. So where you're restricting some foods from the diet. So the growing children, I think they should be exposed to all kinds of foods. It is basically at the age of being an adolescent or a teen, that is a time when their diet changes, where they are moving on to a very unhealthy kind of a fast food or more of an obesogenic lifestyle. So that is the age which needs to be targeted rather than in the age of children. So children should be exposed to all kinds of healthy foods and even like unhealthy foods. I'm not saying like you should be consuming more of the fried foods or something, but consuming more amounts of different kinds of foods will also boost their immunity levels.
Okay. So the next question is, what are the essential micronutrients for the better growth of children?
I think like all the micronutrients are essential to the children because if you take vitamin D deficiency, like nearly 80% of the Indian population, they are vitamin D deficient, and even like B12 deficiency, which is very common. So B12, vitamin D, they are very much essential micronutrients, which have been shown to be the deficiency of which has been shown to be risk factors for the development of various chronic disease outcomes when they become an adult. So these are something that needs to be done regularly because half of the population, they do not even know that they are vitamin D deficient and B12 deficient.
Okay. So what is the difference between lifestyle and dietary change? Because the diet also includes in lifestyle.
Well, lifestyle includes everything. So your behavioral activities, your physical activity levels, your diet, and various other lifestyle like smoking and alcohol consumption. So they all can be categorized as your lifestyle or your environmental contribution. Whereas diet as such, it's just basically whatever you're consuming as such. Yeah.
Okay. So it's been said over generations, the genetic type has been changed based on the lifestyle techniques. Your
I
opinion
don't
on
think,
that?
no, that is not true. You cannot, what do you mean by genetic type? Can you rephrase your question? Because your genes haven't changed. You cannot change your genes. If you're zero copy, you have to remain with zero copies. But if you're two copies, you have to remain with your two copies. You cannot change your type of your copies. But maybe what the question was, maybe more to do with mutation. So there are two types of genetic variations. One is a polymorphism and the other one is a mutation. So mutation is very rare in the population. So where your DNA could have undergone some kind of a spontaneous mutation or any change due to the exposure or due to exposure to chemicals or during the process of evolution. So that is a completely different area of research that has got nothing to do with nutrigenetics, because nutrigenetics is something that deals with your single nucleotide polymorphisms, which are very common in the population, which I was talking about, the 0 copy, 1 copy, and 2 copies.
Okay. So the next question is, what would you think in an underdeveloped country, how can they concentrate on the personalized nutrition when they starve due to already prevailing condition and new COVID crisis as well?
Well, this is more of a question that you should be asking to your Prime Minister, Indian Prime Minister, because personalized nutrition is a research, and what you're talking about is more about the economic burden and the economic conditions of the country. So, but one of them, I mean, as you have raised this question, I would like to mention that as part of the genuine collaboration, the genuine collaboration is not only focusing on implementing nutrigenetic studies in developing countries, but it is also focusing on developing governmental policies to make personalized nutrition free of cost and which can be affordable by everyone. So that is the main target of this project. And I do a lot of workshops and workshop activities, and I conduct several conferences and meetings all over the world, and in order to bring awareness on nutrigenetics and nutrigenomics, because to reach the public with the concept of personalized nutrition, it is very much important that they need to understand what is nutrigenetics. Because a study was done in the US population in 2010, where they wanted to know what is the awareness on nutrigenetics and nutrigenomics. In that survey, they identified that only 6% of the US population, they had some idea about nutrigenetics and nutrigenomics. That was the situation in the US population. So you could imagine what would be the situation in a population like India or any part of developing countries. So in order to bring the awareness, it is very much important. You need to talk more about nutrigenetics than personalized nutrition.
Okay, sir. Next question. What we have to do about the food poisoning, that the food with high nutrition, even if we need to consume it?
What do you mean by that? Food poisoning is completely different and something. I didn't get the question. Can you ask them to rephrase the question? But because food poisoning is because of the spoiled food.
They mean to say that the nutritive value of the food is too high. So in that case, maybe could that poison?
Well, I mean, anything that exceeds the limit is a poison, definitely. It becomes toxic to your body. So every medication that you consume on a long term, it will not have an immediate impact, but on a long term, that will definitely have an impact. And that every medicine that you take has got a side effect. And likewise, every supplement that you take, if you exceed the limit on a daily basis, that will definitely have a long-term impact on your health. Definitely.
Okay, doctor. So next question. How do Nutrigenetics analyze genetic makeup of a person? Is it by studying the inheritance or acquired traits?
You cannot look at the inheritance pattern because single nucleotide polymorphisms, they do not exhibit a specific inheritance pattern because you're looking at all the complex diseases such as obesity, diabetes, and cardiovascular disease, or even cancer. And they are caused by multiple genes and multiple lifestyle factors. It is not one gene. So you cannot look at the inheritance pattern, and there is no pattern. It's basically a complex inheritance pattern. So you cannot decide on based on the inheritance pattern. It is more about the acquired traits, looking at the traits. Like, for example, the traits relating to diabetes, looking at the fasting blood sugar or fasting insulin or BMI or waist circumference. So these are the factors that you will look in terms of doing a nutrigenetic study.
Okay. So nowadays, people are consuming protein powders containing steroids to build up their body. Are there any severe side effects?
Well, this depends on your protein consumption. So how much protein shakes are you consuming, and what is the concentration and the dose of the protein shake that you're consuming? So that depends on what levels of physical activity and workouts you're doing. Because people who are doing very high workouts, they are the individuals who should consume high amounts of protein shake. Because a person who doesn't do any physical activity and consuming more amounts of protein rich foods and consuming more amounts of protein shake, they'll definitely have side effects. And that's exactly what I had shown in my study in the Indonesian population, the Turkish population, where I saw that exceeding the protein content per day, the dietary recommended allowance, that will definitely have an impact on your body. And in terms of, it can also lead to gaining more weight because the protein is high in calories, and at the same time, you need to burn them by doing workout, and so that the protein can be absorbed by the muscle. So it's completely a different concept in terms of using the protein shakes. You need to do workout to burn that amount of consumption. But if you're not doing that and consuming too much of the protein shakes, then that's going to definitely have severe side effects.
Okay. So what will happen to the child if the mother consumes unhealthy nutrition with respect to genetic change? Like what kind of changes or
Yes.
how it will affect the child?
Yeah. So in fact, like we have carried out a study looking at the way we had actually followed up the pregnancy period of the mothers, and this study was done in Indonesian population, where we looked at the first trimester, second trimester, and third trimester, and we also looked at the newborn babies, and we found that the mothers during the third trimester mothers who had very low vitamin D levels, who are vitamin D deficient, they basically gave birth to babies who had very small head circumference. So that study clearly showed, that study was published early this year. And in that study, we were able to show that vitamin D deficiency during the third trimester can result in the newborn anthropometric measurements. So the small head circumference for the babies has been linked with the development of cardiovascular disease in adulthood. So which clearly shows that unhealthy nutrition during pregnancy period will definitely have an impact on the newborn babies.
Okay. So this question is from a participant who is working on air pollution. They want to know how can Nutrigenetics help the people who are affected by air pollution?
Okay, so that's actually a very interesting question. We recently published a paper, it's in press at the moment, which is a study on obesogens. So obesogens are basically chemicals that can induce obesity by affecting your endocrine system. So that can have an impact on that can result in endocrine disruption, affecting your hypothalamic or your brain signaling pathway. So these chemicals are basically available in the air, and which is one of the reasons could be air pollution, and also use of the plastic products. So they can all induce an obesogenic environment for you, which can also have an impact on your gene expression levels. So the reason why you're becoming obese, it is not only because of the genetic factors, but the food that you're consuming and the air that you're inhaling and the products that you're using. So too much of the plastic usage, the plastic has got this bisphenol A, BPA, which can enter into your body. And this bisphenol A chemical, it resembles another hormone, which is called as the estrogen. So estrogen is required for the normal functioning of your body and for carrying out various other factors. So this chemical, when it goes inside the body, it will mimic or it serves as a proxy for estrogen. The cells will assume that this BPA is basically an estrogen, and they'll take up this BPA, and this BPA enters into the nucleus, binds to the DNA, and regulate the gene expression. So if your cells are exposed to this BPA for a long term, and after a while, your gene expression level is being regulated by BPA and not estrogen. So definitely all the air pollution and all these chemicals in the air, they'll definitely have an impact on your gene expression levels, and which is one of the reasons why there is very high prevalence of obesity all over the world, especially in countries where the pollution is very high.
How does the Nutrigenomics work on sports drink?
Well, there are certain genes which are making an individual physically active, and there are certain genes which are making certain individuals physically inactive. So the sports drink has been designed in a way which is actually enhancing the gene expression levels of the genes, which are in a way they are actually making you inactive. So you're boosting the inactive genes to become more active. So that is what they do as part of the nutrigenomics work.
Okay, so this question is interesting. What will you suggest for a person who has no money in his hand to afford your mixed diet?
Yeah, so if you don't have money, I don't know what to suggest for that person, but it all depends on like what kind of diet you want to consume. So uh, again, like I would say that this is more of an economy related question, so I don't want to like suggest anything that is out of this topic, so we'll stick on to this nutrigenetics or nutrigenomics topic. Thank you, sorry about that.
Okay, so can the traditional way of having food change the genetic type of next generation?
Can the traditional way of having foods will change the genetic type of next gen? That's exactly my point. You cannot change your genes. Genes cannot be changed. You're inheriting your genes from different populations, sorry, different generation. So you cannot change your genes at any point. What you can change is only change or modify your dietary pattern, your lifestyle pattern, your physical activity levels. You can change the environment. Moving into a natural environment, that is the one that can prevent you from developing the disease and overcoming the genetic risk. You cannot change your genes.
Okay, so this question is from Mr. Nijan. His sister is 19 years old and she's around 120 kgs. She tried to reduce her weight using different solutions, but she's not successful. So he wants some tip, and he wants to know whether this is due to genetic problem.
Well, I don't want to comment on this because I don't know the other measurements and height of the person and see the concept of like being obese. Just because you're obese doesn't mean that you have a defective metabolism going on inside your body. Even if you're obese, you can still be metabolically stable because the things are changing at the moment. People who are obese can be metabolically stable, and people who are non-obese, they can be metabolically unstable. So the whole concept of obesity is changing now, because obesity, once it used to be considered as a disease, but now obesity, it's just kind of a situation of reducing the weight. So just because you're obese doesn't mean that you're metabolically unstable. So this person, 19-year-old and 120 kgs, if that person is really very high and tall enough, then 120 kgs is actually not too much of a weight. But then, of course, then it depends on the consumption of the diet and the surrounding and various other factors. So there could be a genetic cause as well. So there are children who are extremely and morbidly obese, and those individuals like who have a BMI greater than 40. So there could be a situation of a mutation in a gene. It might not be something to do with the genetic polymorphisms, but it could be something to do with mutations in certain genes like leptin or leptin receptor. Because the leptin gene mutation is very important in the sense, if there is a mutation in the leptin gene, that can prevent you from getting the signal to your brain. If the brain is not getting the signal, then you will end up in eating more and more amounts of food and you gain weight. So there are several factors which are controlling your food intake, and one of which is a leptin protein. So a mutation could be possible. So I have no idea about, like, I haven't seen the clinical reports. So I would suggest that you talk to a doctor or physician or a dietitian or a nutritionist about this.
Okay. So
But one
when
thing
can
to all
we?
the people who are trying to reduce your weight, fasting is definitely not the solution to reduce your weight. Fasting is definitely not. Either you modify your diet and also at the same time have a balance of all the physical activities. These are the only two things that can help you to overcome weight loss. Overcome weight gain.
Okay. So
Yeah.
next question. When can we expect a personalized nutrition-based food and drinks in market?
Well, they're already being like produced in different countries at the moment. So I don't think there's anything in India. Well, yeah, in India, we have like different personalized nutrition centers. They are not products of foods or drinks, but they are providing personalized nutrition diets for individuals based on their different genetic makeup. So if you are interested in knowing about any particular personalized nutrition center, you can drop me an email. I can suggest you some centers.
Okay, this question is regarding the whole COVID situation right now. So they want to know, like, even if a person has a good nutritional diet, good lifestyle, why do they say that the immunity level to recover from COVID is low?
Well, when you say good nutrition diet, it's a good nutrition diet is something that you define as a good nutrition diet. So unless you talk to a nutritionist or a dietitian, you do not know actually what exactly you're consuming. So you might think that what you're consuming is healthy and it's good for your body, but at the end of the day, your DNA is unique, and whatever diet you're consuming has to be personalized. So if your diet is not personalized according to your genetic makeup, then whatever you think is healthy might not be suitable to your body. So you might have a normal BMI, you might be metabolically stable, and there could be various other things like which are actually making you quite healthy. You can feel quite good about your health, but then there are various other factors which are controlling your immunity levels. And that can bring down your immunity levels as well due to the genetic susceptibility and also various other lifestyle factors.
Okay. What could be the mutations for the cause of both diabetes and heart diseases?
So this question is more about the mutation. So as I said, mutations are very rare in the population. That is, they occur at a frequency of less than 1% in the population. Whereas polymorphisms are very common in the population. That is occurring at more than 5% in the population. So when you talk about mutations, they are very rare. So they generally cause very rare diseases. The diabetes and heart diseases are complex diseases which are caused mainly because of polymorphisms. So if a mutation is resulting in diabetes, then it could be an early onset diabetes, which is called as MODY. MODY stands for maturity onset diabetes of the young, which occurs in the age group between 20 to 25. So which is a very crucial age. And that is because of the mutations in certain genes. So that is actually one example of a mutation causing diabetes.
Okay. So the next question, does the natural selection always favor short-term survival rather than long-term health?
See, natural selection and the survival of the fittest and all these concept of evolutionary theories, you cannot apply to the nutrigenetics or the personalized nutrition over here, because the nutrigenetics deals with all the complex diseases. See, for example, if you take obesity or diabetes or cardiovascular disease, if you take diabetes, we know that if you're consuming high amounts of energy dense foods, not doing exercise, you'll get diabetes. That is, your glucose levels increases in your blood. So these are some of the known pathways. And if you become obese, you'll become diabetic. So you know that there are certain pathways or physiological pathways which you know for sure you're going to develop diabetes. But diabetes could also be a result and an outcome of various other factors. There are various multiple factors which are controlling a chronic disease outcome. That's exactly the reason why several people, they respond to a particular kind of anti-diabetic drug, whereas some people, they don't respond to the same drug. So we all have a personalized nutrition approach and also kind of a personalized medicine requirement. And one medicine which can work for one disease cannot work for the other thing. So in terms of looking at selecting individuals based on various other genetic factor or various other factor, you need to think about how this could be personalized at an individual level rather than at the population level. Or at least you need to look at the population subgroups rather than the entire population. So one size fits all approach is gone. Now we're all looking into one size for one individual. So that is what we are all focusing towards at the moment.
Okay. So what is the role of epigenomes in determining obesity gene expression?
Well, epigenetics or epigenomics is completely a different field of research. So what I'm talking over here is more about the genetic approaches, whereas epigenetics is clearly a non-genetic approach. So it is quite different. Genetic changes cannot be changed. You cannot modify your genetic changes. If you're 0 copy, 1 copy, 2 copies, you have to remain with those changes until the last day of your life. Whereas epigenetic changes are reversible. That is, epigenetic changes are nothing but the DNA methylation. So that methylation, the regions in the promoter regions of the genes, that can be reversed. Epigenetic changes can be reversed, whereas the genetic changes cannot be changed. And that's why epigenetics is considered as a non-genetic area of research. And definitely epigenetic changes can have an impact on obesity gene expression, because methylation is the one that determines whether a gene has to be expressed or not expressed.
Okay. So can environmental factors affect FTO gene?
Of course, I think that's exactly my talk focused on, I guess.
Okay. Is environmental pollution in any way related to obesity?
Yeah, I think that was actually part of my answer for air pollution. So I've already answered that question, I think. Pollution and obesity.
Few people eat a lot, but still they remain lean. They do not get fat. How is that possible?
Few people eat a lot, but they still remain lean.
Yes.
Is that your question?
Yes, sir.
Yeah.
So
the main reason, like, well, you have the answer from my talk. So everybody's DNA is quite different. So genetic susceptibility plays a very important role. And the heritability of obesity is basically 40 to 70%. So which means that 40% could be genetics and 60% could be your environment, or the other way around, it could be 70% genetics and 30% environment. So that varies from one population, one ethnic group to another ethnic group. So genetic susceptibility plays a very important role. So some people who might carry a very low genetic risk, maybe that could be the reason why they're still maintaining a very healthy body or a fit body.
Okay, I guess this is the last question to you, Dr.
Yeah.
Vimal.
Yes.
Is having lean fat good for health?
Sorry, what is that? Thin?
Is having lean fat good for health?
Well, I mean, the most interesting aspect about the Asian Indian population is about this concept about the thin fat Indians. So Indians, even though they don't look obese or even though they don't look big, they still have a thin layer of fat in their body, which is predisposing them to develop diabetes. So this concept was developed by Professor Yajnik, and this was published several years ago. And this is a very interesting finding for the Asian Indian. So it's a very unique Asian Indian clinical phenotype, even though they don't have a very high BMI, and still they have that thin layer of fat, which is increasing the predisposition or increasing the risk of becoming diabetic. So definitely, yes, that will have a very big risk.
Okay. Thank you so much, Dr. Vimal Karani, for patiently answering so many questions. I believe there are no further questions to be asked. In that case, over to Mr. Nitish.
Yeah, once again, I thank Dr. Vimal for answering so many questions, and it was a very good session from him. So, coming to the end of the session, I would like to thank Dr. Vimal especially. And this session is also supported by Bionist and Golden Jubilee Biotech Park for Women. And a special thanks goes to Dr. Sudhakar, General Manager, Golden Jubilee Biotech Park for Women, and Dr. Ganesh from Tokai University, Japan. So once again, thank you, Vimal, and thank you, audience, for patiently listening to this session. Hope you all had a great session today.
Yeah, thank you very much.